Okay, our next speaker is Marlien van der Merwe. Marlien's a senior research scientist at the Botanic Gardens, same place as Maurizio. She's passionate about science that guides quality conservation and restoration outcomes. So Marlien's going to talk about representative genetic diversity and quality conservation germplasm collections. Thanks, Marlien.
Find my bearings here. So, as Dave introduced, I'm Marlien van der Merwe, and I would also like to thank the warm welcome we received this morning. I've always felt welcome, made welcome by the traditional owners of the land in Australia. So, let's move on. So, what I'll be presenting today are results from our team that we've been doing, following up on concepts and ideas that is in no way new. I'm starting here with a title from a paper that was published in 1974, so 50 years ago, with this very apt title, Genetic Conservation, Our Evolutionary Responsibility. And I just want to highlight I think it was like the first sentence of the second paragraph in the abstract. Wild species increasingly endangered by loss of habitats will depend on an organized protection for their survival. And I think, I'm not sure how much you can read of that, but the last sentence links up with one of the questions that was earlier about how do we motivate people, and just there, that species of no direct use to man raises questions of responsibility and ethics. So that's just a little bit of, if something's lost, it's lost forever, and it's our responsibility to take care of that. So I'll move on just to go from there. And so I don't think it's difficult at all to persuade this audience that we need to make sure that our germplasm collections, seed collections, seed production areas, living collections, etc., are indeed insurance collections against extinctions. So we don't want to put a bit of seed in a seed bank. That's not going to ensure the survival of that species. We want to do a quality collection from the start. We want to make sure those collections are reservoirs of genetic diversity so that these collections can enable quality restoration or conservation efforts later. And also what I've highlighted there is so that they can also provide quality material for research. Don't want to base your research on one inbred population and then say later that's what a species is doing. So with that in mind, I'll be talking a little bit about what is an insurance collection, what exactly can it be given my background in population genetics. So a genetically representative germplasm collection, I believe, is a collection that will fulfill those criteria. And we can make a genetically representative collection either for a site or for a region or for a species total distribution. And this is our best effort to ensure that these populations or collections can respond to future challenges and other changes. Now, I've got in a speech bubble just a little thought that often I hear from people about germplasm collections. If you talk about, I can't remember how many species are plant species in Australia, but I know New South Wales is just roughly 5,000 or so, and people think, well, how the hell are we supposed to do this? Particularly if every individual is so different. This is impossible. We're never going to do it. I'm going to go home, crawl under my doona. Which I think that's not the way to go. So as a population geneticist, we often use landscape level patterns of genomic diversity to describe the sort of extent of diversity across the landscape. And we use these changes to talk about patterns and evolutionary processes. But one of the things that's been difficult for us is to take these measures and quantify and say, well, based on this information, this is how many individuals capture X amount of diversity. So we've been sitting around and talking about genetic data and think, well, how can we use this data to provide guidelines to people for germplasm collections? So one of the options that comes up is the idea of allelic diversity estimated using single nucleotide polymorphisms. So we can now use with the increase in or the availability of high throughput sequencing and whole genome sequencing, we can really start to look into the genome and really quantify the amount of diversity in an individual or in a population. So I've just to kind of bring it back to nice sort of what is a single nucleotide polymorphism or what is allelic diversity. I've got the 2 homologous chromosomes there, and if you zoom in, you take a little microscope, not really a microscope, but if you go and you see, well, on one chromosome in the exact same position, we've got a nucleotide, which a G, and on the other one, it's an A. So that is an allelic diversity. It's simply the diversity that we can either find within an individual or we can find it between individuals in a population. So ideally, we want to go and capture all that diversity. So I'm just going to walk you through this, because I'm walking you through these basics because I still sometimes have to sit down and think about it. Maybe most of you have got your heads around it. But if we sort of think about a piece of DNA and we look at it, we zoom in on it for 3 different individuals, those sort of stippled, long, I can't remember what you call that shape, a diagonal thing, a thing. Anyway, I forgot, my memory is lost. But if you zoom in on them and you see that here we've got 3 individuals, and if you look at those colors that I tried to represent the allelic variation, you can see all 3 of these individuals are different, but they're made up of the same set of different alleles. The combination of these alleles differ how they are distributed across the individuals. And if you start to look at it, you see that down at the bottom, we've got a homozygote across all those alleles, and then we move up and there's a little bit of a change. So out of those 3 loci, we have the top thing that we can have 6 different alleles, and the individual at the top wins, even though it's just really got 1 different allele from all the others. So based on this information, we can now start to think of how can we look at this to quantify diversity for collections. So we thought, well, this is pretty simple. We can think of the idea of when you do field work and you do a accumulation plot for how many species in an area. This is what we would like to get to with our genetic data. So we want to plot our alleles as we increase our individuals and see where's that nice little circle that I drew there, that sweet spot where as we increase individuals, we're not really getting any more allelic variation. So if we can get a germplasm collection that is in that spot, we've captured a large amount of diversity. We haven't captured all the genotypic variation and recombinations and things, but with those different alleles, we can potentially in the future recreate this over time, of course, many, many millions of years for some species. But based on this information, I comfortably feel that we don't have to crawl under the doona and go and cry, because based on this, we can make small quality collections that can go into seed banks or conservation collections, and we capture a large amount of diversity. So what I've done to go and test this is to go and sample a lot of seed from multiple species, from multiple families, grow them up, genotype them, and then sit in front of the computer, thanks to other people that's mostly done this for me, and then plot how we increase our diversity and see, can we get to that sweet spot, that plateau, or does it just keep going up? And some of this data for 2 of the species I won't talk about. The 2 hakeas are published in the paper. You can follow that QR code if you feel like it. And then I'm just going to talk about some results we're getting. So these are what I would like to refer to as these accumulation of alleles in a seed collection. So it's not an individual seed that I genotyped. I genotyped the individual that survived, so that the seed that actually germinated and grew up. So what we found is that we do get that nice plateau if we plot the allelic diversity like that. But what we do find is that we don't necessarily find that, we only find that depending on your collection strategy. So for this specific species, you can see that the sort of darker blue at the top is where we collected from 3 populations, and those lighter yellow is when the collections only came from one site or one population. So we can repeat the same thing for another species. So I've just got another acacia next to it. 2 acacias, they look quite similar, they do similar things, they make similar looking flowers, but they've got very similar, different patterns in this way. And we also found this when we looked at the more population level genetic diversity patterns. But what we see here is again that for multiple populations you get more diversity than you would get for a single population. So we can make quality collections if we visit multiple populations. You don't have to go and over-harvest from those populations, but you can make small collections, and there's enough seed left in the population. You fulfilled your scientific license. You didn't collect more than you're supposed to, and but you can still capture a lot of diversity with these relatively small collections. So I know what you're thinking is that, but hey, I'm not going to drive all the way out there and collect 45 seeds. I'm going to collect way more. So I thought I just want to have a little look what happens when we do collections at a site, just so you can get an idea for that as well. So what I have here is we did a 5 minutes. I think I'm more than halfway, so I won't have to talk too fast. I can speed it up. So what we have here in the orange is when we collect from multiple maternal lines, while the blue is the same number of seed on the plot, but from a single maternal line. And this is all common sense. If you're going to think, I'm collecting from different mums, I'm going to get more diversity. And you can see the nice pattern there. And if we then do the same and we look across different species, what we find is that we get different patterns across different sites. We don't get the same amount of diversity at a site or per maternal line. And we also don't get the same patterns across species. So this might make you think this is a bit more complicated. What am I going to do? But I think we've established that visit multiple sites, collect from multiple maternal lines, and you're going to capture more diversity than if you go to a single site and pool them all. Which brings me to the next baby of mine at the moment. Oh, no, conclusion. But I think I can jump that one because I've already kind of concluded. But on here, I have number 3 there. Keep your maternal lines separate. So why do I want you to keep your maternal lines separate when you do seed collections? Because it's a lot of work to put them into different envelopes and then have to process them later. But we've got, just from these 2 examples, I've got quite a lot of information that shows us on this plot is just when we germinated everything for this, we found that different mums don't all germinate at the same rate and they don't all have the same number of viable seed. So if you kept them separate, you would know actually in the end my seed collection only represents 2 or 6 or 10 or 12 or whatever number of maternal lines. So that already gives you an idea of how many maternal lines are represented in your seed collection. The other thing, of course, that is that if you want to use the seed, you want to make sure you don't increase the process of inbreeding. And if you kept your maternal lines separate, that's a way easier way of making sure that if you're going to use this seed, you're going to avoid that. So finally, just to conclude again, keep your maternal lines separate, and these make much better seed collections. There's many more reasons for keeping maternal lines separate. You can go and, I think we gave 10 or 11 reasons in a little paper we published last year. Follow the QR code. And then just to summarize, I think I've said or I've summarized everything, so I can make a bit of time up here for somebody. But the information here is relevant to when we set up seed production areas, and it's very relevant when we do conservation collections and conservation gardens, is to remember to keep your maternal lines separate and make sure your seed collections are genetically diverse. And then finally, I'd like to thank you all. I'd like to thank everybody that helped with collections. And there's a conference coming up next year about seed science in Australia. Thanks.
Thanks, Marlien.